Buenos días a todos y todas. Mi nombre es Kniel, soy la directora ejecutiva de ILSI Mesoamérica y es para nosotros y para el Comité de Inocuidad de los Alimentos de nuestra organización un honor darle la bienvenida a esta cuarta sesión de cápsulas que hemos estado organizando, que el comité ha estado organizando. Y el día de hoy vamos a hablar sobre esos agentes, parásitos, protozoarios que son de interés en la industria alimentaria. Tenemos el honor de contar con la participación el día de hoy de la doctora Kniel de la Universidad de Delaware y como moderador del doctor y profesor Óscar Acosta, director del Centro Nacional de Tecnología de Alimentos de la Universidad de Costa Rica, quien nos hará el honor hoy de ser el moderador y de realizar las traducciones técnicas de las preguntas que ustedes quieran hacerle a la doctora Kniel el día de hoy. Así que bienvenidos a ambos. Welcome, Dr. Kalmia, and thank you for joining us and your support to realize this session today. Y gracias, Oscar, por estar con nosotros. Bienvenidos. Voy a hacer una presentación muy rápida de lo que es ILSI. Recordemos que somos una organización global sin fines de lucro, que trabajamos de forma con el más alto nivel de integridad científica para proveer ciencia que ayude y que impacte la salud pública, el bienestar y el desarrollo sostenible de manera positiva. Somos una organización tripartita donde los científicos convergen para trabajar en temas específicos y generar programas o proyectos que de alguna manera generen y comuniquen ciencia en todo el mundo. Como pueden ver, existimos prácticamente en todo el planeta y eso nos permite pues tener acceso a científicos de todo el planeta en temas de interés para la región. Esta es nuestra misión y nuestra visión para el siglo XXI. Es muy importante para nosotros pues ser para toda la población de México, Centroamérica y el Caribe pues aquella que genere y que esté comprometida con los principios globales de integridad científica, que reúna expertos de los diferentes sectores para generar esas actividades que con transparencia generen y publiquen y sean esa plataforma de convergencia de expertos para impactar positivamente estos temas. Y además, pues esperamos para el año, estamos trabajando para que en el año 2030 seamos un referente impulsor de ese alto nivel científico en la región, en temas relativos, por supuesto, a lo que nos compete, que es a la salud pública, el bienestar y el desarrollo sostenible. Como siempre, hacemos una declaración de exención de responsabilidad y es que las opiniones que se presenten hoy son las del presentador, experto científico, y no reflejan las de ILSI, no lo hacen en nombre de ILSI Mesoamérica ni de la Federación, pero además todas, y siempre agradecemos el que todas las personas que participan como expertos lo hacen de manera voluntaria, han tenido total libertad para preparar sus conferencias y tampoco han recibido remuneración alguna. Por eso siempre le damos las gracias a nuestros expertos, los que participan en nuestros eventos, porque lo hacen de manera voluntaria y nos dan no solo el conocimiento, sino también experiencias y su sabiduría y nos la comparten. Por eso siempre les pedimos que tomen mucha atención de este evento. De inmediato, pues le voy a dar la palabra al doctor Óscar, que nos va a presentar a la doctora Kniel, que nos acompaña el día de hoy. Muchas gracias, doctor Óscar, y bienvenido a esta sesión.
Gracias, Jania. Muy buenos días. Agradezco enormemente el honor de poder estar aquí con ustedes el día de hoy para moderar esta sesión técnica. Bueno, usted amablemente ya realizó una introducción. Me presento nuevamente. Soy Óscar Acosta, director del Centro Nacional de Ciencia y Tecnología de Alimentos. Y como tal, represento con muchísimo gusto tanto al CITA como a la Universidad de Costa Rica. A continuación, lo que voy a hacer, si espero estén de acuerdo, voy a hacer una breve introducción de nuestra presentadora. Lo voy a hacer en inglés por respeto a la presencia de ella y luego probablemente haga una breve presentación en español sobre su trayectoria. So, Dr. Kniel is a professor of the Department of Animal and Food Sciences at the University of Delaware, specifically the Department of Animal and Food Sciences at the College of Agriculture and Natural Resources. She completed her graduate degree at Virginia Tech, focusing on cell biology and microbiology of foodborne pathogens. Her teaching and responsibilities include courses on foodborne disease, food micro, food systems, and food security. Her research includes understanding mechanisms of environmental persistence by bacteria, protozoa, and viruses. And actually, our topic today is going to be entitled Protozoa and Parasites of Concern for Food Safety, a topic in which she has quite an extensive experience. She also serves as co-chair of the One Health Unique Strength Program and directs the Center for Environmental and Wastewater Epidemiological Research. She is co-author of the textbook Food Micro and Introduction, more than 100 scientific publications, and more than 170 published abstracts for presentations. In 2015, she was awarded the University of Delaware Outstanding Teaching and Advising Award and the Elmer Marth Outstanding Educator Award by the International Association of Food Protection, IAFP. In 2020, she was awarded the University of Delaware's Outstanding Researcher Award. And in 2022, she was awarded the IAFP Maurice Weber Laboratorian Award for Distinguished Laboratory Contributions. She is active with the Institute of Food Technologists, with the American Society for Microbiology, and also with the International Association of Food Protection, where she was elected in the board in 2015 and served as president in 2020. So with that, I'm going to just do, like I said, a very brief introduction in Spanish, and then I'll give Calmia the opportunity to start her presentation. La doctora Kniel es una profesora catedrática en el Departamento de Ciencias de Alimentos y Animal en la Universidad de Delaware. Ella realizó estudios de grado y posgrado en Virginia Tech. Sus responsabilidades de investigación y docencia están organizadas alrededor principalmente de lo que es microbiología de alimentos e inocuidad alimentaria. También cumple labores administrativas de dirección y codirección de algunas organizaciones de gran relevancia para la Universidad de Delaware. Es coautora de más de 100 publicaciones científicas y más de 170 abstracts de presentaciones en eventos internacionales y ha sido honrada con una serie importante de premios en organizaciones de reconocido impacto y nivel internacional, incluyendo el Instituto de Tecnólogos de Alimentos y también la Asociación Internacional de Protección de Alimentos. El título de la presentación que la doctora Kniel va a compartir con nosotros el día de hoy es Parásitos, protozoarios de importancia en la inocuidad alimentaria. So thank you very much, Calmia, for being here with us, and it's all yours. Thank you.
Okay, thank you so much. It's such a pleasure to be here. Let's see, does that look okay?
That looks perfect, thank you.
Thank you. Really, thank you very much to ILSI and ILSI Mesoamérica for the pleasure and the privilege of being here with you today to talk a little bit about protozoan and parasites of concern. So just to share with you where I'm sitting today, I am centered in the sort of middle Atlantic area of the United States, here nestled about 2 hours between Washington, D.C. to the south and 2 hours north of New York City, in case you've been to those areas. And this is the College of Agriculture and Natural Resources at the University of Delaware. And so I'm sitting in this building, and then over to the right, you can see kind of our main campus area. So it's a pleasure again to be able to talk to you today about kind of an overview and some specific comments and thoughts on certain foodborne protozoa that are of continuing importance when it comes to food safety. We'll spend a little more time on the organism Cyclospora cayetanensis, in particular talking about what we've learned from various outbreaks, and then discuss what we're all trying to do as we move forward with awareness and prevention in mind when it comes to these protozoa. So protozoa are single-celled microorganisms that divide within their host. They come in a variety of shapes and sizes. I believe that our attendees today have various backgrounds, and so some of you may have more of a veterinary background and may be familiar with different protozoa. Certainly Giardia, Cryptosporidium, Toxoplasma, they're very important when it comes to some of our zoonotic diseases as well. And then of course we have Microsporidia there on the right-hand side, which is an interesting protozoa that actually has chitin and seems to be maybe more like a fungi than a protozoa. There are many others as well that are important in terms of public health, but these are the ones that we really think about when it comes to more food transmission. These are very large microbes compared to many of the others that we talk about when it comes to food safety. So on this slide, you can see the bacteria, which are about 1 micron in size, the virus, which of course are nanometers in size, and then we have Cryptosporidium, which is about 4 to 5 microns in size when we look at the size of the oocyst, And then cyclospora is maybe about 10 to 12, or even maybe a little bit larger in terms of the micrometer size there. And Giardia can be much larger when we look at the trophozoites. So when we line these organisms up and think about their potential to wreak havoc on our food safety systems, right, we really are looking at these specific species: Cryptosporidium, Cyclospora, Toxoplasma, somewhat Giardia, although we don't seem to get Giardia into the food system or across the food chains like we do with some of the others. And there are several things about these organisms that inherently make them greater concerns. One is their very low infectious dose, anywhere from 30 oocysts, perhaps when it comes to Cryptosporidium, up to 100 or so, or maybe even more. We don't know very much, unfortunately, about Cyclospora cayetanensis, and we'll talk about those knowledge gaps in just a few minutes. The other thing that makes these organisms very critical when it comes to food safety is that they are resistant to normal levels of our chemical disinfectants that we rely on when it comes to cleaning and sanitizing. So here on this slide, you can see, again, some of these images of the oocysts that are transmitted by Cyclospora and Cryptosporidium. Again, we have these challenges in particular when we think of fresh produce and the produce industry supply chain. These oocysts are really quite a challenge when it comes to cleaning and sanitizing, as well as education and training of our harvesting crew and our workers, who of course are really critical to the food industry as a whole. So these organisms, again, are, we see low infectious dose with them, robust nature, their resistance to washing and removal and chlorine. But then that's coupled with a lack of awareness, right? We talk a lot about Salmonella and Listeria. We don't talk so much necessarily about Cryptosporidium or Toxoplasma. We don't understand their transmission as well when it comes to transfer from a bin to a fruit or a leaf, or to a conveyor belt. We have lots of questions there. We haven't been able to do some of the studies that we need to do, and that's because of the lack of standardized methods for detection and identification. I'll talk about that briefly today. There's also somewhat of a lack of understanding of their life cycles, in particular for Cyclospora. And when we think about how do we detect these across our food supply chain, we're really going back to those methods where they may not be robust enough to be able to detect low levels that could be there in our food supply chain that may lead to disease. So let's start by having a look at Cryptosporidium parvum. And I'm going to use Cryptosporidium here as our example of talking about one of the challenges of studying these organisms is that they have a complex life cycle. And here you can see what happens over on the left corner side when an oocyst is ingested. It goes through what's called excistation, where you have this sporozoite that exists, that comes out of that oocyst. The sporozoite then can infect the human intestine, and it undergoes a series of asexual reproductive cycles. And so you may end up with a rather quick colonization of intestinal cells. And then following those cycles of asexual reproduction, the organism undergoes sexual reproduction as well, where you can see it differentiates into a microgamete and a macrogamete that then turn into these microgametocytes. You have then formation of a zygote that then is shed as the oocysts out into the environment. What's really unique about Cryptosporidium, and you can see that here in these labeled J and K on the left-hand side, is that about 20% of the oocysts that are formed inside the human host, are what's called thin-walled. And those thin-walled oocysts rupture inside the host and lead to this auto-infection cycle. This is quite dangerous for individuals who are severely immunocompromised and are not able to mount the cellular immune response that's needed to be able to overcome this organism. So this is a very simplified look at this life cycle that comes from the Centers for Disease Control and Prevention in the United States, right? The oocysts that are shed into the environment are those thick-walled oocysts. They then contaminate water or food. The individuals there are shown in a swimming pool. We do see quite a number of outbreaks that are associated with recreational water, and that's because Cryptosporidium is completely resistant to the levels of chlorine that we use in swimming pools. So those thick-walled oocysts are ingested by the host, and then after they replicate in the intestine, the oocysts are shed already infectious. So they are immediately infectious as soon as they enter the environment. Now, over time, certainly scientists have developed advanced and much better diagnostics when it comes to detection of Cryptosporidium. It started off using microscopy. Of course, that's sort of the basis of all detection systems. We still can use microscopy. Here on the right-hand side, you can see that top image there is looking for some cyclospora oocysts and perhaps a vegetable wash matrix. And then on the bottom, you can actually stain oocysts, and they will take up an acid-fast staining, especially for Cryptosporidium there. But now we've moved on to more rapid diagnostics like immunoassay kits, but predominantly PCR, polymerase chain reaction, is used to be able to detect Cryptosporidium, and real-time PCR is used so that we can quantify how many oocysts there might be in a sample. Although there's a lot of trouble with this when it comes to our challenging food matrices, and including environmental samples like water or soil. So when it comes to clinical diagnosis of protozoa, I thought it would just have a brief discussion here on the use of these culture-independent tests. This is something that I think is really interesting when we think about attribution and burden of disease when it comes to food safety for all of our protozoa. Depending on the clinical testing that's being done, we may see more and more cases that are being detected clinically in hospitals, and then we have a difficult time of attributing those to specific foods or specific outbreaks. We see that certainly for cyclospora. We also see that for Cryptosporidium, and on this slide that again comes from the US CDC, you can see that there was an increase in the use of these culture-independent methods for Cryptosporidium. We also see that only culture-independent methods are used really for cyclospora detection, and those went into place at the same time that we started to see this increase in the number of cases. So it's very interesting how those align with each other. And we do in the United States actually see more cryptosporidiosis, the disease caused by Cryptosporidium. You can see this brown kind of dotted line there going up towards the end. Many of those cases have to do with either unpasteurized juices or recreational water. And I think that around the world, we see smaller outbreaks that tend to be kind of regularly occurring. So the infectious dose for Cryptosporidium parvum or Cryptosporidium hominis, which are the two organisms that can infect humans, is about 100 oocysts or so. Could be a little less, could be a little bit more. Cryptosporidium parvum is the zoonotic species that can infect humans. It also infects ruminants like deer and cattle. Cryptosporidium hominis seems to just infect humans. And really anyone can get sick after ingesting Cryptosporidium. Immunocompetent individuals may still have a fairly severe diarrhea that is self-limiting though after about 7 days or so. And immunocompromised individuals unfortunately can get a very severe or wasting diarrhea that may last longer. And we do see that there is some success with an FDA-approved treatment in the US, as well as HIV patients can actually, or individuals with HIV, can see some reduction in Cryptosporidium infections with the heart therapy, and that's because those protease inhibitors can actually work on the proteases that are important for excistation of the organism, and you can see that here in this picture. So again, Cryptosporidium is really unique because it's immediately infectious when it's shed in the environment, and while these organisms are larger than bacteria, they're still fairly small, and so it is difficult to filter them out successfully using a water filter, but they can be filtered. so they can be retained in some of the filters, which is great news. So if we look globally when it comes to distribution of cryptosporidium, this is where I think we need a conversation globally in terms of surveillance, because it's very difficult to ascertain the global burden of disease when it comes to cryptosporidium, right? When we look at our inadequate detection methods in the United States as well as in other countries, it's difficult to test for these organisms in foods or in environmental matrices. It's not that I do not suggest that we all go out and start testing our food. That is certainly a waste of money. Rather, it's important that we look at good agricultural practices and things like that. But it's important that we try to understand really globally what's happening, because it's likely that there's an underestimation of protozoa really around the world. And as we are seeing our immunocompromised populations grow across the world, we really do want to try to better understand what's happening in terms of the potential for cryptosporidium. So I will now transfer to the next organism. So this is sort of like a trip as we go through these different organisms. The next one is Toxoplasma gondii. Some of you may be quite familiar with Toxoplasma. In the United States, it's actually the second leading cause of death due to foodborne illness. And I say that even though we really don't have the best reporting, it's likely underreported. And also, when we think about transmission, it's quite difficult to understand routes of transmission. Certainly we know how it occurs, but we don't know exactly where our numbers are coming from. So Toxoplasma gondii is the protozoan parasite that causes the disease toxoplasmosis. This is actually an infection that has a very high prevalence worldwide. Individuals can be asymptomatic, or may have very mild symptoms. This is actually not a gastrointestinal illness. The organisms insist into our skeletal muscles or other muscles, and so may actually cause a flu-like symptom as our immune system is responding to those infections. Infection can be obtained in 3 different ways. That's ingesting the oocysts that are shed only by the definitive host, which is the cat. And that's either can be contaminating on fruits or vegetables or in water. There were, in the literature, there's a couple papers that suggest that oocysts can be transmitted through air when there's many cats that are around a barn, for example, but we don't see that happening epidemiologically very frequently. Or you can ingest tissue cysts, which is what's shown here on the slide on this picture. And that would be an undercooked meat, most likely pork, lamb, horse meat, boar, bear, or cougar. We don't see as much transmission with cattle. And then also, tragically, there's congenital transmission, where a pregnant woman acquires the infection during pregnancy, and then that infection can be transmitted to the fetus and actually can cause hydrocephalus or water on the brain, and then can also lead to fatalities of that fetus. And that's, tragic, and certainly something that is important that we have good education of our physicians so that they can have those conversations with women. So if we look at the life cycle here, and then on the left-hand side, on the top, there is a sporulated oocyst. So that's the oocyst that's infectious. And then the tissue cyst is what forms in the animal host or the human host even. And that's a different life stage. Those are very different than the life stages that are transmitted in the oocyst. So if we start here on this life cycle, and there you can see kind of the 3 transmission routes that I just mentioned. We can have transmission from a pregnant woman to the fetus, or you can have transmission through ingesting oocysts. This is why women are often recommended not to deal with cat feces while they're pregnant. Although if you are already seropositive before you become pregnant, you're actually protected from that infection. And so I think it's important that women are knowledgeable about their if they're seropositive or not prior to getting pregnant. We don't do those testing very frequently in the United States, but it's my understanding in some countries around the world they do those more often. And if you look at all of these other animals that are potentially zoonotic hosts, right, they play an important role in terms of transmission. It's very interesting how little we know around the world about infection through meat, right? It's been, and this I provide you with a lot of text here. I apologize that there's so much text, but I thought it would be better to put it up here because I think this is a very unique paper that looks at this question about disease burden. What can we do to try to reduce the burden that Toxoplasma gondii has on our disability adjusted life years? So you may be familiar with this word DALY, disability adjusted life years. So when we look at the impact that foodborne illness has on us, it's not just our time away from work or the time that we're sick, not just the impact necessarily on the recalls or on the food system, but we really want to think about the totality of the cost of illness. And here we can look at this burden of Toxoplasma gondii, and it's estimated that there are tens of thousands of cases, and it's that's taking away thousands of our of our quality of life days. And this is something that's happening globally, as you can see here, globally when we look at what's happening in South America, Middle East countries. This is a really important pathogen that we need to know more about. But unfortunately, we just don't really have a very good set of data when it comes to looking at attribution. So this image, which comes from this paper here to the right, is where it all starts, where the oocysts start. So the oocysts can be shed by a cat, a feral cat, a cat that's living outside, who has the ability or chance of eating a rat or a bird that's already infected and ingests these tissue cysts. The tissue cysts then can rupture when they're in the cat, and they lead to the infection of these cells. And this part only happens within the cat, which is known as the definitive host. And then cats shed the oocysts in their feces that can get out into the environment. So again, once those oocysts are ingested, right, they can be ingested through contaminated water or fruits and vegetables and transmitted to people that way, or they could be picked up in the pasture and transmitted to other animals that then get tissue cysts. So there's a few educational aspects here, right? We want to think about how we are washing fresh produce. We want to think about the role of cats in the environment. Although that seems to be fairly minimal, what is less well understood is the role of contaminated meat. In some of the big studies in the US where scientists have looked for tissue cysts, they really don't find them. And so that's quite intriguing. So does that mean we're really ingesting oocysts then rather than the tissue cysts? But tissue cysts can also be transferred by handling under a raw meat or meat juices. And so we have the need then for proper education to make sure that people are aware that this is a potential route of transmission. So this is a slide I wanted to just share, because while we're looking at how we raise animals, in many parts of the North, South, Central America, we raise pigs like this picture here in the center, where we are raising them on a concrete floor. We have very good biosecurity systems. We're monitoring what goes in and out. We're monitoring who goes in and out. And that's done so that we can maintain the health and welfare of those animals. Now, if you can see up on some of these other pictures, and here we have chickens or poultry as well as swine, there is an increasing desire to raise animals in these types of conditions. These are the conditions that we need to be a bit more concerned about when we think about protozoa that can be transmitted this way. In parts of the world where we're seeing more and more animals raised this way, we're also seeing more cases of toxoplasmosis as well as trichinella, which is a helminth that can be transmitted through pigs. So something to keep in mind how we're raising our animals and how important biosecurity is as well. So I believe that some of you may be familiar with Angiostrongylis. cantonensis, which is known as the rat lungworm. I thought I would mention it here. It causes eosinophilic meningitis, is prevalent in parts of the southeast of Asia, as well as tropical Pacific islands. And interestingly, we seem to, in the United States, now be having a few cases of this. In fact, we just had some cases last week that came up into the state of Georgia. And so now we're seeing a little more rigor being put on discussing this organism and thinking about how it may be able to contaminate some areas of fresh produce or juice production. So I thought it was worth mentioning here because it seems to be more on people's radar. This is what happened just last week. And so people are really trying to be more aware, even though that this roundworm, not a protozoa, I'm sorry, stepping out of my lane for a few minutes, but it seems to be present. Okay, so let's spend the rest of the time talking about Cyclospora. So Cyclospora cayetanensis is probably the most important protozoan parasite that we see these days. I would say up and down through Middle and Central America, up into North America. They don't seem to have the cases in Europe like we do, but there's a growing need to better understand this organism when it comes to the science. Remember that these are not bacteria, so protozoa do not grow outside of the host. They only grow once they're ingested by the human host. And actually, Cyclospora is not zoonotic. It's only shed by humans as well. And once it's shed in the environment, it has to actually become a mature oocyst, so it needs some time to sporulate. And so it takes a little bit of time. There are numerous questions that remain and so many challenges about this organism. We actually don't have an animal model or a cell culture laboratory model that we can use for this. I mean, it's very difficult to obtain oocysts. So here are some images of the Cyclospora oocyst. You can see on the top left is actually the oocyst that is unsporulated, so that would not be infectious. After about 10 to 14 days in the environment, it becomes infectious, and that's where the blue arrow is pointing to. And the oocyst actually contains sporocysts that contain sporozoites, and the sporozoites are what infects the cell, similar to those that we saw for Cryptosporidium. And so there is this time frame that's needed after these enter the environment, and that brings about a lot of questions when it comes to transmission across the food chain. What happens during that time frame if we ingest oocysts that are not mature, that are not fully sporulated? We probably don't see any illness. Also, the cold chain is being questioned in terms of how does it retard or slow that process of sporulation. So these are all things that we need to know more about. This is the life cycle, again, showing you an image from the US Centers for Disease Control and Prevention. The oocysts that are sporulated or mature are ingested by the human, and those undergo a series of asexual reproductive cycles followed by sexual reproduction that then leads to this oocyst that can be shed in the feces. That oocyst is unsporulated, so it has to stay in the environment for about 7 to 10 days. Maybe that depends, though, depending on the temperature outside and the conditions. We do see transmission and outbreaks that occur with Cyclospora with various herbs and leafy greens in particular in the United States. So this is a disease that can affect both immunocompetent and immunocompromised individuals. Some people can actually be infected and remain asymptomatic. Unfortunately, this disease can last for quite some time. So it may be a self-limiting diarrhea after about 5 to 10 days, or it may go on for a longer period of time of up to 6 weeks. And again, it's a watery diarrhea, but then followed by quite sort of the typical gastrointestinal symptoms, loss of appetite, bloating, cramping, nausea, fatigue, right? So really an unpleasant disease to have. There is treatment, though, that's available, but mostly these are sulfa drugs. So individuals who have allergies to sulfa drugs will not be able to take these. So again, we need more more research done there as well. And if we look at how long it takes to be able to detect a cyclospora infection, it really takes about 3 to 6 weeks here in the United States. And this is actually using one of those culture independent methods. So in the US, in our clinical hospitals, many, if not all of the hospitals have this instrument that's called a BioFire film array detection unit. And this BioFire panel for gastrointestinal illnesses actually detects many bacteria and viruses, as well as parasites. And it's a small panel, about the size of a cell phone or so, and it can detect the nucleic acid of these different organisms. Now, these organisms are shed in very high amounts in human feces, and so it's easier to detect them. We can't use the same system when it comes to food or environmental detection. So testing and detection are really difficult right now when we think about what's going on in terms of food. It would be great if we would be able to have a reliable, quick assay that we could go out and do some environmental monitoring, or we would be able to use even in outbreak situations. But at this time, really, most of the methods are primarily restricted to research purposes. I'm going to mention some FDA methods in a minute, and there are ISO methods that are being further developed and tested for cyclospora. But it's a very difficult situation. This is certainly one of those big knowledge gaps. The methodology continues to evolve. There's validation for various food matrices. And I think detection from food, scientists do very well. It's the detection from environmental matrices that is very tricky, right? Pulling these oocysts out of soil, comparing detection of these oocysts of cyclospora specific in a water, for example, the environmental water that's shown here that might be used for irrigation. There are lots of wildlife that are in that water, and so there are related species to cyclospora. So how do we know which controls to be able to include to make sure that we're really detecting only cyclospora? That's become a tricky question here, especially in the United States. So this is, there are 3 different methods that have been used and published by the US Food and Drug Administration in the Bacteriological Analytical Manual, known as the BAM. Mostly now, Chapter 19B is being used for detection of cyclospora in fresh produce. And then there's also 19C, which uses a dead-end ultrafiltration method for the detection of cyclospora from agricultural water. And so that actually can be quite useful, where you can collect water and then bring it to a lab and run it through the filter. And then you can test for the organism using real-time PCR in the backflush that comes out of that filter. And so this is a method that's being further studied on all different types of environmental water samples. And I think that this is really important that we continue to see this further development. And also, I think the ISO method will probably be very similar to something like this, and will certainly be real-time PCR based. There are ISO methods that are available for detection of Cryptosporidium and Toxoplasma and Giardia, and those, of course, are excellent methods. So I'd like to talk a little bit about what we've learned from some of the key outbreaks when it comes to Cyclospora cayetanensis. So from the 1990s into the early 2000s, we saw numerous outbreaks that occurred, mostly from produce that was grown outside of the United States and imported in, although that wasn't completely well documented in some of the recent outbreaks. But if we look at some of the information that came to us through the early 2000s, right, these were often berries or snow peas, herbs, or various lettuces. And some of these outbreaks, the product was something that people did not recognize eating. For example, it could have been a sun-dried tomato vinaigrette that included some basil, and you might not know that. And then also the raspberry coulis or the sauce that's around a cake, for example, and you wouldn't remember eating that. Those recalls in terms of human memory are very tricky sometimes when it comes to outbreak investigation. So that, you know, can make traceback very difficult. Also, it was not until just recently that we've had more advanced molecular diagnostics and the ability to run real-time PCR to be able, or qPCR, to be able to detect these organisms at the low levels at which that they are in these products. And then if we look to after 2013 and forward, and these are all in the United States, these outbreaks that have happened that have been linked very specifically to cilantro or basil or bagged lettuce or even the broccoli that was in a vegetable tray, while many others are unknown. And it's interesting to me that the use of that BioFire panel coincides at the same time that we start to see from 2013 onward many of these large numbers of cases of cyclosporiasis in the US. We would have thousands of cases, including those of unknown origin that were not linked specifically to an outbreak. So what is the origin of some of these cases when they can't be linked specifically to an outbreak or specifically to a certain food commodity? So beginning in 2018, the CDC added a new means of classifying these cases, and they call this domestically acquired cases of cyclosporiasis. And that's in individuals who showed no international travel that was associated with these cases. And here you can see the numbers for the year is the first number there, 2018 through 2023. And that was as of this summer. And then you can see there are many hospitalizations that go along with these cases of illness. And what's interesting, and I haven't mentioned yet, is that cyclosporiasis is predominantly a seasonal illness in the US, where we see it mostly in our summer months. And so perhaps the weather has something to do with the sporulation rate in the environment. I think that there's something there that should be better studied as well. So another interesting point of data that we have goes along with an herb sampling plan that was, or surveillance plan that was done by the FDA. This is one of, they have multiple assignments like this. This is the microbiological surveillance sampling for fresh herbs. And here they're actually looking at Salmonella, E. coli O157H7, as well as other sugar toxin producing E. coli, and then Cyclospora cayetanensis. And here you can see with both domestic herbs as well as imported herbs that there's just very few positive samples that are being detected, right? So roughly the same percentages really between domestic herbs and imported herbs when you look at how many tests were done, and that's kind of across the board for all of those. So fairly interesting there. And I think that they had paused this assignment during the COVID pandemic, and now they are restarting this assignment. So I think they're going to continue some of this testing. So in 2023, the CDC, the FDA, and state and local partners investigated an outbreak of cyclosporiasis cases that occurred in the southern United States, in the states of Georgia and Alabama. These were linked to raw broccoli that actually included about 20 illnesses. People reported that they had eaten or consumed broccoli prior to when they got sick. The FDA was not able to identify specific producers of the imported broccoli as the source of the outbreak. The outbreak is over at this time, at least seems to be over. So they're not sure exactly if the broccoli was, if this outbreak can be attributed to the broccoli, and they were never able to detect anything. And so this is actually a screenshot from those investigations, and you can see there were, as of actually just a couple weeks ago, these were still labeled as active investigations and ongoing. You can see some of the cases there. We have just a couple hundred cases, which is good since we've sometimes had thousands of cases. but it's still lots of questions about where are these oocysts coming from in terms of the environment. So there really is a clear need for standard method performance requirements. There is an AOAC working group, and that actually has members from North America, from Central America, and South America that are working to inform these standards that are being developed. We need to understand more about the organism. In terms of writing these methods, it's always important that we talk about the definitions, and so we know exactly what we want to call an herb, for example, and what should be tested. We have to have good controls. We have to know where surrogates can be used for research, which I think is really important, so that we can provide guidance when it comes to ways to reduce contamination by this organism in the environment. And then there's also this need for descriptions of processes and detection controls. So we can have continuity, so we can use these methods in my lab and in your lab, right, and across the globe. I think that's really a critical piece. So when we think about how do we move forward with this knowledge of foodborne protozoa and how they're there in the environment, we want to be able to prevent disease. We also want to do some future research. We want to know where those knowledge gaps are. We want to be able to try to understand better, I think, the global attribution for all of these organisms, right? This is a very complex problem, but when it comes down to it, we certainly can look to reduce cases with more simple solutions, right? We can all decrease risks, right? We can try to engage in surveillance when we can, but perhaps we look at waters, right? We want our water to be fit for purpose. How are we using water when it comes to fresh produce or across the food supply chain? What type of water are we using? Because if we think that oocysts could certainly be present in water systems, that's going to be a really important question. How are we using that water? Also, we want to consider sewage spills and sewage treatment. It's possible that if we have ineffective sewage treatment, that oocysts can be making their way into water systems that then can be used in food chains. We also want to think about the role of portable toilets, especially in the U.S. and in many of the ranches in Mexico and in Central America. We want to think about where are those being emptied, how are those being handled, are there recommendations in terms of those practices. And in terms of industry controls, certainly attention to hand washing remains critical, good sanitation of restrooms and portable toilets. We want to avoid the use of sewage sludge or managing spills when they happen. It's important that controls against human fecal contamination are implemented really across the supply chain. So while these are not bacteria, we can use some of the same controls in terms of good hand washing and good hygiene, and then cleaning and sanitation, although these organisms will not be inactivated by those sanitizers. The persistence of cyclospora in the environment remains a really big question, right? We have hundreds, if not thousands of cases every year. Where is it coming from? How is it? Is there an unknown reservoir, for example? Scientists have lots of questions. The triggers for sporulation are really not well understood. Is that why it's a seasonal disease here in the US? Because you need to have those right temperatures. How does the acidity of a fruit factor into that? We've done some experiments in my lab where I've questioned that, berries, we might have different interactions of the oocysts with the berries than you might have with the lettuce leaf, for example. So I think we can all come to this content place of really focusing on prevention measures, right? And since many of these organisms are transmitted by people, we want to think about people, right? Focusing on training across the food supply chain. I'm showing you fresh produce here, but it's always important across the entire supply chain that we have good education and good training and open communication so that workers can say to upper management, I'm not feeling well today. I should not be at work. Those kinds of communication channels are really critical. So I think it's important to train employees in their native language. I apologize that I'm not speaking in Spanish today. I wish that I could. I think it's also important to train farm workers on how to use the facilities that are there, because sometimes there's cultural questions that come up. We see that in the US for sure, and I think that we see that around the globe. Importance to share the why about hand washing, right? Not just to do it, but to explain why it's so important, right? I think those why questions are really important, that we answer those when it comes to training. Also talk about the symptoms that are associated with different diseases. And again, that helps explain the why. Ensure that there's supervisory oversight when it comes to worker health and hygiene and sanitation. Ensure that tools can be properly cleaned and sanitized before and after your workday. I always work with my growers to talk about the labeling of tools, maybe different colored tools for different activities. And then cleaning and maintenance of portable toilets is very important. Developing a plan and having corrective actions in place before challenges occur if it comes to leakages or spills. We have quite a bit of flooding here that occurs, especially this time of year where we have tropical storms that are coming off the coast. And so there's often flooding events, and it's best that we have plans for those situations that might happen before they happen. So I just want to say thank you again for this opportunity. It's really a pleasure to be here to speak with you. I'm happy to answer any questions. And also, this is my email. Please reach out to me if you have any questions or comments. And thank you.
Thank you very much. That was a very interesting, informative presentation, I have to say. So thank you very much. We do have a couple of questions. I actually have a couple more, if you don't mind. So I'm going to start with the ones from audience. And what I want to do is I'm going to read them in Spanish. I'm going to then try to translate, and then I will try to interpret your answer and then give it in Spanish. Okay, so in Spanish. La pregunta es: ¿recomendaría muestrear el agua de una planta de deshuese de bovinos y cerdos con la finalidad de detectar parásitos a pesar de tener un esquema de limpieza y desinfección estricto? So in English, the question is, would you recommend sampling water from a beef and pork processing plant, specifically a deboning plant, to detect parasites, even though the facilities might have strict cleaning and sanitizing protocols?
Yes, that's a great question. So I would not recommend testing that for parasites. I think if you're using your fecal indicators, for example, E. coli, that could tell you if you have intestinal or some type of a contamination. I know in some places they're still sampling diaphragm tissue, for example, to look for trichinella, or you may be looking at mussels for specific life stages of other parasites, but I'm not sure there's a ton of merit to be testing water for those parasites. It's too expensive. I think there's other indicators that could help give you better information.
Thank you very much. La respuesta, bueno, primero indicó que es una excelente pregunta y es su experiencia que no es necesario, que es mejor usar indicadores de contaminación fecal, como por ejemplo E. coli, que es más confiable. Podrían haber otros indicadores, por ejemplo, hizo algunos ejemplos del muestreo para trichinela o realizar muestreos de tejido muscular también para detectar algunos otros microorganismos. Y finalmente, la pregunta, aunque es buena, considera que no tiene mérito realizar ese muestreo de las aguas para buscar los parásitos específicos, porque podría ser caro y resultaría quizás más eficiente utilizar otros indicadores. Okay, so I'm going to move to the second question. Again, I'm going to read it in Spanish and then translate to English. En español, ¿qué tipo de desinfectantes son recomendables para una buena desinfección en la industria alimenticia para ayudar a controlar esto fuera de las buenas prácticas en general? So in English is, what type of sanitizing would be recommended for a food industry to help control parasites outside of, well, taking into account good practices in general?
Yes, that's a great question. So unfortunately, most of the parasites, and the protozoa in particular, they are resistant to normal levels of chlorine. There is better disinfection when it comes to some of the peroxyacetic acid chemicals. For example, hydrogen peroxide can be effective against even cryptosporidium. UV light is also can be effective, and ozone. So for example, in treatment of water, UV and ozone can be very effective, but if there's a great concern, then maybe a non-chlorine based, more the peracetic acid or UV or ozone, but even those have some limited effectiveness.
Thank you very much. Bueno, menciona que además es una muy buena pregunta y que desafortunadamente los parásitos, específicamente los que estamos analizando el día de hoy, que son protozoos, tienen una resistencia al cloro particular. Entonces se recomienda lo que es el ácido peroxiacético, por ejemplo, lo que es la luz ultravioleta o lo que es el ozono. Sin embargo, estos sistemas de desinfección también tienen unas amplias limitaciones. So I'm gonna insert my question right here because it kind of relates to the previous one. So basically, I had a question that you kind of answered towards the end of the presentation, but I'm going to throw it out anyways. So instead of decontamination and interventions is basically training and prevention, right? What industries and governments can do right now. Is that actually what you would say?
Yes, I think that prevention is certainly the key, because once we have these oocysts present, it's very difficult. It can be very difficult to get rid of them.
Ok, perfecto. Mi pregunta, porque me tomé la libertad de lanzarla en este momento, tenía que ver con los comentarios que la doctora Kniel realizó al final de su presentación, que ella indica que debería enfocarse básicamente el control en aspectos preventivos y también de capacitación en las empresas. Entonces mi pregunta era precisamente relacionada con la anterior sobre los sistemas de desinfección o las sustancias que se utilizan para la desinfección. Es que más bien en lugar de realizar intervenciones que procuran la descontaminación, en realidad, por lo menos a la fecha, de las iniciativas que podrían tener mayor mérito son la capacitación y la prevención de la contaminación. Ok, tenemos una pregunta adicional. En productos congelados como brócoli, ¿cuál es la incidencia de la presencia de protozoarios? So the next question is, in frozen products such as broccoli, for example, what is the incidence, the presence of protozoa?
That's a great question. So we have not seen epidemiologically frozen product linked to illness, though these organisms, so cryptosporidium, can survive some degree of freezing in soil, but we don't really have a good understanding of what happens on fruits and vegetables. And I'll also add that tissue cysts of toxoplasma actually can be inactivated by freezing. So that's a good thing.
Thank you very much. Bueno, nuevamente es una buena pregunta. Desde el punto de vista epidemiológico, no hay suficiente información que permita vincular productos congelados con estos peligros, en este caso los parásitos protozoarios. Hay evidencia que, por ejemplo, algunos de ellos pueden sobrevivir durante la congelación. Sin embargo, no podría asumirse como una técnica correcta de descontaminación. Y aquí yo voy a aprovechar para lanzar una pregunta y la voy a hacer en inglés. So, based on that, I had another question for you that had to deal with technology. So we know that parasites in general are sensitive to thermal processes, right? And then you mentioned that some technologies, for example, UV light, sanitizers, which again, chlorine-based may not be as effective. Is there any innovative technologies that you're aware of that are being worked on in different parts of the world, I'm assuming, that might help, at least in the following years, kind of have like a decontamination step for products, which I know is challenging because you cannot freeze and then thaw, for example, many of these products, or even heat them, right? So what is your knowledge in this topic?
Oh, this is a wonderful question. I think our continued development around the world in these non-thermal processing technologies is very exciting. So my lab has actually done some work with chlorine dioxide and shown that that can be effective against oocysts. Cold plasma has some effectiveness. Of course, that's more difficult to be able to bring to a food supply chain, but chlorine dioxide certainly has its merits and is probably close to being commercialized. So I think that those two are very promising, but UV is also effective. You just have to reduce the shadowing of the product.
Thank you very much. Mi pregunta tenía que ver con la aplicación de tecnología para el control de estos microorganismos, el cual, bueno, por la información que nos han brindado en la charla, sabemos que no es una etapa que en este momento podamos contar con una aplicabilidad, digamos, absoluta. Pero básicamente mi pregunta es si existe alguna tecnología que tal vez no esté implementada actualmente, pero pueda ser utilizada en el futuro para el control de estos contaminantes. Y la respuesta es que existe investigación utilizando, por ejemplo, lo que es el dióxido de cloro, lo que es el plasma frío y lo que es la luz ultravioleta. Sin embargo, ellas deberían utilizarse, por supuesto, con cautela por las limitaciones que presentan. Ok, tenemos una pregunta adicional. ¿Qué se debería hacer? So you guys threw a curveball at me because it's in English, so now I will have to translate to Spanish. ¿Qué se debería hacer con brócoli crudo? Lavarlo con detergente y luego realizar un enjuague. Se podría utilizar yodo, por ejemplo. Entonces sería más bien una pregunta con respecto a los consumidores. Por ejemplo, también lo que son las fresas en México, si la desinfección con yodo, perdón, sería suficiente. So this question is more, I'm assuming, is more oriented towards consumers. What can people do at home? And they're giving some examples. For example, using iodine for sanitizing at home, because should people just wash really well and then rinse, or should people use iodine or bleach, or like, I don't know if it's the objective of the question, but I'm assuming that is more oriented towards what people can do at home.
So I think it's important to note that consumers should wash their fresh fruits and vegetables only with water, not using soap or bleach or anything like that, because that can be dangerous and has risks of other unintended effects and unintended consequences. It's also important to wash your hands, of course, but when dealing with meats and being sure to take the meat and to not, let's see, often we have packaging materials, right, that contain juices, so you try to avoid touching those, and then cooking with a meat thermometer is always recommended to the right temperature. I wouldn't recommend at home using any of those other chemicals because of the unintended consequences and the lack of inactivation of the organism.
Perfecto. Ok. Bueno, la recomendación sería no necesariamente realizar una desinfección, porque se ha evidenciado que no hay un efecto particular de esos agentes sobre los microorganismos. Entonces la recomendación es una buena aplicación de prácticas de lavado que sean adecuadas, o sea, la remoción mecánica que sea importante. Se podrían tener efectos no deseados, por ejemplo, del uso inadecuado de esos desinfectantes, sea cloro o sea yodo, como la persona que nos brinda la pregunta. Y si bien es cierto, la pregunta está enfocada en brócoli y en fresas, si no me equivoco, también se hace la observación que en el caso, por ejemplo, de los productos cárnicos se debe tener muchísima precaución con lo que es el manejo, tanto del material como de los productos de desecho, por ejemplo, los jugos que permanecen en el material de empaque o el mismo material de empaque, y también el tema de la correcta cocción, temperatura de cocción más bien de esos alimentos. Y ahora pasaremos a la última pregunta para terminar con tiempo la sesión. So this is the final question because I think we're running just out of time. What is the incidence of these microorganisms in aromatic herbs that can be sold for infusions, for leaves, I'm assuming, or products that come from the ground basically and that are used, not eaten per se, but are used for infusing and obtaining a drink?
Interesting question. So I don't know the incidence because we don't do a lot of testing. But if we look at that testing that the FDA has done on imported and herbs grown in the US, it's very low. So I think the incidence is very low. But they are found on cilantro and basil and herbs that grow fairly close to the ground. But I think it depends also on the leaf. So cilantro has little hairs and the oocysts can stick there better. So I think it's probably very low, depending on the herb.
Thank you very much. La pregunta se refiere al peligro de estos microorganismos en hierbas aromáticas que pueden ser utilizadas para infusión. Y la respuesta es que aunque no hay suficientes datos para lanzar una conclusión sobre este tema, aparentemente la incidencia es baja. Si bien es cierto, se reporta incidencia en alimentos como por ejemplo culantro o albahaca, esa incidencia depende mucho también de la fisiología del vegetal, en este caso de hoja. Entonces la respuesta es que, bueno, no se descarta la posibilidad de que ocurran, pero por lo menos la incidencia que se ha reportado es bastante baja. Con esto creo que estamos con el tiempo. Jania, yo le agradezco muchísimo la invitación a colaborar en esta sesión tan interesante y le cedo entonces la palabra. Gracias.
Thank you very much, Dr. Calmia, for joining us, and Dr. Acosta for being here as a moderator. We are so happy to have a lot of people today in this session, even if it is in English, but many people stay with us all the time. Thank you very much, Dr. Calmia, and I hope to see you again in our next session about this topic in 2024. So thank you very much for you and for Professor Acosta too. Bueno, muchas gracias a todos por haber estado con nosotros. Queremos recordarles que esta semana tenemos muchos eventos y el mes todavía de septiembre y de octubre es muy importante para nosotros que nos acompañen. Hoy mismo por la tarde vamos a tener un evento en el marco de la celebración del Día Internacional de las Ciencias. Esperamos que algunos de ustedes nos puedan acompañar. Es a las 2 de la tarde y vamos a hablar sobre vocaciones científicas en personas jóvenes que estamos haciendo en la región. Además, a partir de la semana que viene vamos a iniciar una serie de seminarios que serán 4 sobre lo que es la alfabetización en alimentación y nutrición, con 4 sesiones donde vamos a ir hablando sobre qué es, por qué es tan complejo, qué hemos aprendido de las estrategias que ya existen a nivel latinoamericano y por último, cómo trabajar, cómo usar las tecnologías y la digitalización para comunicar todos los temas que tienen que ver con nutrición y alimentación. Tenemos expertos de toda Latinoamérica que nos van a acompañar en estas sesiones y esperamos pues que vayan a nuestra página web y se registren. Además, seguimos con nuestras sesiones de trabajo en aquellos temas que estamos desarrollando sobre los alimentos, los nutrientes y la salud. Seguimos con proteínas, todavía esta semana que viene nos faltan 2 sesiones y la de la próxima semana es muy interesante porque es sobre innovación y vamos a tener 5 expertas en este tema extraordinario sobre temas proteínas vegetales, proteínas alternativas, así que también les invitamos a todos a registrarse en este seminario y participar con nosotros. Y bueno, me parece que quedan 2 más. Mañana tenemos un evento también en la Universidad de Costa Rica en el marco de lo que es la pérdida y desperdicio de alimentos, la concientización para disminuir este problema. Recuerden que estamos desperdiciando alrededor del 30% de los alimentos que se producen. Es muy importante que aprendamos a manejar los alimentos y a desperdiciar la menor cantidad posible. Y por último, pues ya para el mes de noviembre vamos a tener otra cápsula de este comité de inocuidad, una cápsula donde vamos a hablar de todo el tema de los avances en las intervenciones específicamente antilisteria en quesos frescos que son tan comunes en la región mesoamericana. Igual pueden registrarse en nuestra página web. Ahí también van a ver, tenemos un enlace especial ahora para todo lo que tiene que ver con inocuidad de los alimentos. Estamos subiendo videos de expertos especialmente para los consumidores. Es un trabajo que el comité decidió hacer, de empezar a comunicar ciencia y temas de mucha importancia para las personas. Ya estamos trabajando en una infografía, en una serie de infografías que vamos a desarrollar para Navidad. De manera tal que les vamos a colaborar para que ustedes aprendan a seleccionar, preparar y utilizar los sobrantes de los alimentos en las fiestas en Navidad. Así que pues estamos llenos de eventos como pueden ver y bueno, en nuestras redes sociales pueden encontrar los detalles para que se registren y nos acompañen. Una vez más, once again, thank you very much, Dr. Kniel, for being here with us and thank you, Professor Acosta, for your great talk today. Thank you and have a good day. See you.
Good day, thank you.
Gracias, Oscar. Chao.